Welcome to our extra credit video. This is a showcase of two DNA extraction labs, one of which is the extraction of DNA from strawberries, the other of which is the extraction of DNA from broccoli. Now, shall we begin? Material used. This material used is dish soap, which breaks down the lipid bilayers of strawberry cells. Next are the plastic bags, which will be used to hold the mixture of strawberry and other materials. Next is a fork, which will mix solutions. Next is a 1/4 measuring cup, which is used to measure the salt. Next is a popsicle stick, which is replaced by a skewer later, which is used to stir the DNA out of the alcohol in the end. And then another important ingredient, 70% isopropyl alcohol, which is used so that the DNA does not dissolve in the liquid. These are the strawberries in which the DNA will be extracted from. Next is the strainer, which removes the hard parts of the strawberries from the mixture. After is the salt, which breaks up the proteins that hold the nucleic acids together. Last is a cup of water, which make up part of the extract solution with the soap, which will be made now. Soap is added to the water and is mixed. The strawberries and the solution of soap and water is added to the plastic bag. Along with salt. This is then mashed and mixed together. The soap will break down the membrane and lipid bilayers, and then the salt will break up the proteins which keep the nucleic acids together. The solution is then strained into a cup, removing the clumps of strawberries. The isopropyl alcohol, which allows the DNA to clump because the DNA is not soluble in the alcohol, is then added. The alcohol is slid down the side of the cup. After 5 minutes, the DNA is visible and can be extracted.
A few moments later.
The skewer gathers the DNA by twirling it, and it can be lifted up when it is on the skewer. This is the DNA that we have extracted. Now, onto the broccoli extraction lab. The cups will hold the solutions. The alcohol will again prevent the DNA from dissolving. The skewers, again, will be used to gather the DNA at the end. The soap used to break the membrane and lipid bilayers of the broccoli. This is the broccoli mix, which contains soft broccoli parts and water. It was pre-made because of the need for a blender. The strainer will remove the hard parts and thick parts of the broccoli from the solution. The spoon will mix. The broccoli with water is then poured into a cup through the strainer, removing the thick parts. Dish soap was also added into the solution. The solution with the soap had to settle for 5 minutes. After that, the alcohol was then given another 5 minutes for the DNA to appear.
Eventually.
After the skewer stirred the DNA and pulled it out, this is the amount of DNA that we got. This is a side view of the cup. As you can see, there is still some DNA there. And that concludes the presentation. In this experiment, we learned the uses of household items by applying them to science uses.